Hello everybody, this is Dr. Anita Srivastav and today I am going to talk about the microbial growth. So basically the topics that will be covered during this lecture will be the bacterial reproduction, bacterial growth curve, factors affecting microbial growth, measurement of cell growth and continuous culture systems. So basically, if we talk about the growth, about any growth, so growth is defined as the increase in size of an organism. And when we talk about the microbial growth, it is not just the increase in size, rather it is increase in cell number. So normal growth and microbial growth. So it usually has a difference of this factor. Basically, this is increase in cell number, not increase in cell size, which we usually talk about the growth. When microbes are fed with nutrients under favorable environmental factors, they actually become metabolically active. That means every microorganism needs some environmental factors, some favorable factors where they can divide and they can be metabolically active, means they can increase their cellular components. So when we talk about the microbes, we all know that bacteria are prokaryotes and most prokaryotes, they reproduce by binary fission. So here binary fission, basically we will be talking about the binary fission. So now, as I was telling you that all the bacteria are prokaryotes, single-celled prokaryotes. So when we talk about the prokaryotes, that means we break this word prokaryote. So prokaryote is pro and karyotes. Pro is primitive, karyotes is nucleus. And these are single-celled animals, whereas eukaryotes means true, nucleus. So basically the differentiation is being done on the difference in the nucleus. And when we talk about the binary fission means the reproduction that is taking place in bacteria through the binary fission. So the growth which we will talk, it is actually defined as increase in cellular components. Increase in cellular components that leads to rise in cell number by binary fission. So actually, how does this process of binary fission takes place? So here I would be using the highlighter. So here we can see that this is the process of binary fission. Initially, a parent cell, a young cell. So basically, it starts for the preparation of the cell division by first enlarging its cell wall, its cell membrane and its overall volume. So here we can see in this young cell, there is a cell wall, cell membrane and elongated nucleoid. Now the septum, this septum I am talking about. So this septum, it begins to grow and the nucleoid divides. Here we can say that this is the nucleoid, but here as the septum increases growing, the nucleoid it divides and the septum completely, it is totally in the position to divide it completely in the cell. So after the complete formation of the septum through the cell center, we can see that there it becomes two different cells, means transverse septum becomes complete. where it can be seen that the cell is going to divide. So now after this step, we can see that it results in two separate daughter cells. So this way the process of binary fission takes place. So this is the binary fission. Now we have to talk about the growth, the microbial growth in terms of the generation time. So first of all, let us understand what is this generation time. Basically, generation time is the time taken to double the population. It's doubling of population in a specific time. So here we can see that generation time is time interval upon number of generations. And by solving this formula, we can say that generation time G is equals to T by N. So now the thing to understand here is how this N equals to this has come. So what we have to understand about the formation of this equation, and how does this equation has come. So first of all, we should understand that whenever we culture any media, so normally there might be some number of cells, but initially we consider that at time t the initial cell number is 1, but actually the number of cells is not 1. Normally there are thousands of cells which are being inoculated. So we can say that N naught is the initial population number, N naught is the initial population number, and N is the final number. So N is equals to N naught into 2 to the power of n, where n is the final number of cells, n naught is the initial number of cells, into 2 to the power of n means the generations, the number of generations in time t. So then on solving this equation by adding the logarithm, so after the solving this n equals to n naught into 2 to the power of n, the formation of this 3.3 log 10 and log 10 and 0 will come actually. So if you all want, I will discuss this in detail about this equation, or about this total formula which we are getting to calculate the generation time. Now we will talk something about the growth curve after the bacterial reproduction. So bacterial curve actually, it consists of 4 phases. We have the lag phase, log phase, stationary phase, and the death phase. Actually this is a graph that is plotted between the time and the number of cells, logarithmics of the number of cells. So in the lag phase, no increase in cell number takes place. Why? Because the reason for no cell division or the increase in number results because initially the cell needs to adapt the environmental conditions as well as the medium. So lag phase is the time actually where cell prepares themselves to synthesize new components, ribosomes, and essential cofactors. Then comes the log phase, and log phase is also known as the exponential phase, where this is the main phase where the cell exponentially divides and double at regular intervals with a balanced growth, as well as the growth rate is constant. Now after the log phase, the cells enters into this stationary phase. So in stationary phase, we can see that there are number of viable cells which remains constant. So now the reason for these viable cells to remain constant, because the, the carbon source, the nutrition, the nutrient is becomes in the limitation state, and it this stationary phase, it results as a balance between the cell division and cell death. This thing. Then stationary phase is followed by the death phase. In the death phase, actually the cells becomes deprived of any nutrient. One reason in the death phase, that is the cell becomes, some cells die. The reason is first I told you that they becomes deprived of the nutrient. The other reason is actually the buildup of the toxic waste products. The buildup of toxic waste product means during fermentation, during any run. Suppose for example during the fermentation reactions, some acids also form, and those acids actually does not provide the favorable or the optimum growth conditions for the cells, so they enters into the death phase. Now we will talk about the factors that affect the microbial growth. There are factors which affect the microbial growth are solute and water activity, halophiles, we have the pH, temperature, and oxygen concentration. So when we talk about the solute and water activity, osmotolerant. Osmotolerant means osmo is osmolarity, tolerant means tolerance basically. So those microorganisms which have the capacity or whichever are able to grow on wide ranges of water activity and osmotic concentration, they are actually osmotolerant. Few examples are Staphylococcus aureus, whereas Saccharomyces rauxi. Then the other factor which affects the microbial growth are halophiles. Halophiles, if we break this term or understand this term, halo, salt files is always loving. So halophiles have the ability to grow and tolerate under the saline conditions, that is high concentration of sodium chloride. That means they can optimally grow under the saline conditions. And the examples are Halobacterium, Dunaliella. Now the other factor which affects the growth is the temperature. It is one of the most important factors that defines the microbial growth. So basically, if we classify or we categorize the microorganisms on the basis of the growth depending upon the temperature, they are the psychrophiles, psychotrophs, mesophiles, thermophiles, and hyperthermophiles. So here in psychrophiles, we can see that psychrophiles, they optimally grow between 0 to 10 degrees centigrade. Means there are, and even in this graph we can see that even the growth starts below 0 also. They can grow. It's not that below 0 it's 0, rate of growth is 0. No, they are increasing below 0 also, and the optimum growth takes place around 10 degrees. Whereas we can say psychrotrophs, they have the optimum growth temperature of 20 to 25 degrees centigrade. Then mesophiles have the optimum growth between 20 to 25 degrees centigrade. Almost all the microorganisms fall under this category basically. Then comes the thermophiles. Thermo itself is telling that they love the high temperature. So basically thermophiles have the optimum growth between 55 to 65 degrees centigrade, and here we can see the optimum temperature for their optimum growth takes place at the temperature of 60 degrees centigrade. Then comes the other class, that is the hyperthermophiles. So hyperthermo means they are thermophiles but above than the 65 degrees centigrade. So the temperature range between optimum growth temperature range between 80 to 100 degrees centigrade, and we can see that the optimum they are growing around 95 degrees centigrade. So these are different kinds of microorganisms favoring different temperatures for their optimum growth. Now we'll talk about the pH. So pH, every microorganisms varies for their growth in different pH, and there are three classes which we can see are acidophile, neutrophile and alkalophile. Those microorganisms which love to grow in acidic mediums are acidophile, which finds favorable to grow in neutral medium, that is neutrophile, and we have the alkalophile. So in acidophile microorganisms, those are acidophile, the growth optimum between 0 to 5.5. And examples are sulfobolus, microphilus, ferroplas, aconitum, cynidium, candarium also. Neutrophils are categorized between 5.5 to 8 pH. They show their optimum growth. And the most known by bacteria, Escherichia E. coli, and we have Euglena and Paramecium also, who loves the neutrophilic conditions to grow. Then the alkalophile growth optimum between pH 8.5 and 11.5. Examples are Bacillus, Alkalophilus and Natronobacterium. So here in this graph we can see the growth rate versus pH, and the pH scale is showing the acidophile, neutrophile and alkalophile. Now the other factor which is very important for the growth of the microorganisms is oxygen concentration. The growth of the bacteria depends upon the oxygen concentration. So there are certain conditions, means some bacteria, they are obligate anaerobe, facultative anaerobe, aerotolerant anaerobe, obligate anaerobe and microaerophile. So if we talk about this first category, the obligate aerobe, means they are completely dependent on atmospheric oxygen for the growth. Examples are Micrococcus luteus, Pseudomonas, Mycobacterium, mold protestant fungi. So obligate aerobe means they cannot grow in absence of oxygen. Then comes the facultative anaerobe. One more thing about the obligate aerobe, like if we culture the microorganisms under the static conditions, actually most of the oxygen is present on the surface. So once they utilize that oxygen, then it becomes difficult to get moved to dissolve oxygen until we supply some aeration, some agitation to it. Then comes the facultative anaerobe. Facultative anaerobes, they actually don't require oxygen for growth, but they can also grow in oxygen. That means they can grow under aerobic as well as anaerobic conditions, and the examples are E. coli, Enterococcus, Saccharomyces cerevisiae. Aerotolerant anaerobe, they grow equally well in presence or absence of oxygen. Example is Streptococcus pyogenes. Obligate anaerobe, they actually does not require any oxygen, and when they are supplied with oxygen, it becomes toxic and dies in its presence. Examples are the Clostridium, Bacteroides, Methanobacterium, Treponema, Agilis, Microaerophile. Then they require oxygen below 2 to 10% for growth and is damaged by atmospheric oxygen. So microaerophilic means they only require a small amount of oxygen. Micro is very small. The example are Campylobacter, Spirillum, volitans, Treponema and Palladium. Now we will talk about the measurement of the microbial growth. So after the growth, the cell numbers are to be measured. So the measurement of cell number can basically the measurement of the growth can be done by the measurement of the cell numbers, measurement of the cell mass, whereas the other is the cell activity. So if we talk about the measurement of the cell numbers, so the measurement of cell numbers, first it can be done directly by the microscopy, and the device used for counting or for the measurement of these cells are Petroff, Hauser counting chamber and hemocytometers. They are special slides which have certain depth, some grooves, some depth of known amount of known size, and the sample is placed from these on these counting chamber, and they are basically used for the prokaryotes, whereas hemocytometers, they are used for prokaryotes as well as the eukaryotes. So these slides, they can be elaboratively seen under the microscope only. They have, like when we talk about the hemocytometers, so hemocytometers, when we will observe it under the microscope, we can see there are a number of squares. Means if we take the volume, then the volume we can see that it is 9 millimeter square. It has almost 16 cells in one corner square, and in the middle square it has around 25 squares. So it can clearly be seen under the microscope. Then the cell numbers are counted by the electronic particle counter, that is the Coulter counter and the flow cytometry. So recently flow cytometry is being used for counting the cells. Coulter counter has two electrodes, and the number of cells are counted when an electrical resistance is developed around the electrodes and when a sample is placed into it, and there we can electrically count the number of cells. Then measurement of cell mass. Measurement of cell mass depends upon the microbial dry weight and spectrophotometry. These are the two ways where we can measure the cell mass. So microbial dry weight. Microbial dry weight we can measure like after a fermentation process. The cells are centrifuged, they are washed, they are dried in an oven. Then we can calculate the microbial dry weight. And spectrophotometry. Spectrophotometry, we all know that it depends upon the Lambert law and the amount of light absorbed by any solution is directly proportional to the concentration of the solution. Here we will talk about the turbidity basically in case of microorganisms. Then we have the cell activity. Indirectly by relating the degree of biochemical activity to the size of the population. Now we will talk about the continuous culture of microorganisms. So basically up till now what we were talking about the microbial, about the bacterial growth, about the growth curve, about the bacterial reproduction, and about certain like we have discussed the conditions. So mostly we were talking about a batch culture. Now we are talking about this continuous culture of microorganisms. So now what is the basic difference about between the batch culture and the continuous culture of microorganisms? Batch culture, in batch culture, we usually supply the nutrients once. Then the product is taken only after the complete fermentation process. Whereas in continuous culture, from one end we are continuously supplying the feed, and at the other end means when we are supplying the feed, that is the inlet, and at the other end continuous removal of the media with the microorganisms are taking place. So a continuous culture system is a microbial population that can be maintained in the exponential phase and at a constant biomass concentrations for extended periods. So here we can see that the number of cells formed is equal to the number of cells removed. That means the under this culture system the cells remains in a steady state. And there actually one factor that is limiting is the nutrient limitation over here. So under nutrient limitation, the microorganisms can grow well and have a good growth potential. Two types of continuous culture systems are chemostat and turbidostat. When we talk about the chemostat, it is designed in such a way that the rate of feed is equal to the rate of removal of medium containing microorganisms. So, means what I told you, that the rate of feed is equal to the removal of the medium containing microorganisms. Second, the rate of nutrient exchange is expressed as dilution rate, comma, the flow of medium per unit of time over the volume of cultured vessel. Means the flow of medium that is taking place in a culture vessel is the dilution rate. Means the flow rate by volume of the vessel will determine the dilution rate. Now the advantages of the continuous culture systems are they are useful and constant supply of cells in exponential phase and annual growth rate, and the microbes grow at a very low nutrient level. This is actually the structure we were talking about, the continuous culture system. In a vessel we have a fresh medium, then this is the culture medium. So from the vessel this medium is supplied into the culture medium, and here we have the air filters, control valve is there, and we control this flow rate by the pumps. And here also a pump will be there which will control the flow, outlet flow, and here we have the product taking place as well as the microorganism. So the number of cells formed over here are removed. Those who show that initially the culture will be in the steady state. Then after the chemostat we will talk about the turbidostat. So turbidostat, they measures the absorbance or turbidity of the culture in the growth vessel using the photocell. So a photocell is being used for measuring the absorbance. Here, as it is quite different from the continuous culture system. Here the dilution rate varies rather than remaining constant. Here, in the case of continuous system, the dilution rate was constant. And all the culture medium contains all nutrient indexes, whereas there was limitations nutrient. Means nutrient was limited. It operates at higher dilution rate. So these systems are essential for research in many areas, understanding the interaction between the microbial under natural environmental resembling like freshwater lakes or ponds. The continuous system is used in food and industrial microbiology. So we can say that these basically the continuous systems, they are used at large scale in the food and industrial microbiology because the production of the enzymes and all the products, they are taking place under the continuous culture system. So it is very useful at the large scale. So I think with this we can end with the growth, and uh, uh, for any queries we can have some